Okay, good morning to everyone, and first of all, thank you to Philips for the kind invitation. And so, okay, so let's start my talk. During my talk, I'll try to give you some examples of how MR can be used in the clinics to help clinicians in making the right diagnosis in patients with neurodegenerative conditions, but also in clinical research in order to better understand the pathophysiology of these conditions. I like to start by the end of my presentation, so with some acknowledgement here, the list of people working with me at the Neuroimaging Research Unit, Professor Filippi, other people here. I'm a neurologist, so here are other neurologists, but also bioengineers, neuropsychologists, and neuroscientists. And of course, we collaborate with the Department of Neuroradiology, so with the neuroradiologists. And just to give you the framework of our institute, at the Department of Neuroradiology, we have 4 Philips scanners and 2 other Philips scanners at our radiology department. So I'm going to show you our findings obtained mainly using the 3T research scanner at our institute. Another couple of background slides. First of all, which are the neurodegenerative conditions we are interested in? Many neurodegenerative conditions, but mainly dementia, so Alzheimer's disease, and other conditions like frontotemporal dementia, but also movement disorders, including Parkinson's disease and amyotrophic lateral sclerosis. All these conditions are characterized by the accumulation of disease-specific proteins in the CNS of these patients, and also by a typical characteristic of these conditions, which is that pathological changes accumulate over time in a stereotypic pattern, in a stereotypic topographical pattern. So the idea is to use MR not only to make the right picture at the initial phase of the disease, but also to try to follow the progression of the pathology in these patients. So which are the MR tools we use in our lab? First of all, we look at the atrophy patterns using T1-weighted images. And here, just an example, you can see a pattern of atrophy in patients with Alzheimer's disease. But we also obtain diffusion tensor imaging in order to assess the contribution of wild matter damage in these conditions, using both voxel-wise analysis but also chartography. We are also interested in looking at function in our patients, and we used to obtain resting state function MRI, looking at specific networks which are known to be associated with these conditions, such as the Default-Mode network, for instance, but also looking more generally at the connectivity of the whole brain using more advanced graph analysis and connectomics approaches. So using 1 hour scan, we can obtain images in our patients to look at different aspects associated with the disease, from the gray matter involvement to the white matter involvement to the connectivity abnormalities. So first of all, the first question is whether we can use these approaches to improve diagnosis. Here you can see an example of this issue. For instance, obtaining a nice T1 weight, 3D T1 weight image from your patient. You can use both visual ratings but also VBM approaches to look at specific patterns of atrophy in specific regions. And in less than 10 minutes, you can apply 6 scales, which are very helpful for the clinician For instance, if you compare the accuracy of the diagnosis using these scales with the diagnosis obtained using more standard clinical approaches, the sensitivity and specificity is very high and is highly associated not only with the clinical diagnosis of the patient, but also with the underlying pathology, which is an important issue in patients with these conditions. We also try to improve more the accuracy of diagnosis, combining the quantitative measures you can obtain using gray matter analysis and white matter analysis and machine learning approaches. Here an example in amyotrophic lateral sclerosis, applying machine learning approaches developed by our engineers like Silvia Basaglia and Camilla Cividini. They are here in the room. So we combine the measures obtained using 3D scan and this mathematical algorithm, and we improve the diagnosis of ALS, amyotrophic lateral sclerosis, not only versus healthy controls, which is an easy task for a clinician, but importantly, compare with the so-called ALS mimics, so other clinical conditions that can mimic ALS in the initial phase of the disease. And then another example, in Alzheimer's disease patients, we combine the information you can obtain using a 3D T1 weight image with a deep learning approach. And for instance, in this paper, we show that using just one T1 weight image without any other information about your patient, you can differentiate between patients with mild cognitive impairment, so those who are in the initial phase of Alzheimer's disease, who are going to convert to over dementia, compared with those who are not going to have a clinical diagnosis within 2 years. So a nice example of the combination between very detailed information you can obtain using MR, mathematical approaches, but also the clinical practice. We also ask whether white matter can contribute to the improving of the diagnosis in the clinical practice. And here are 3 examples from our lab when we where we combine information about gray matter atrophy and white matter damage in differentiating many neurodegenerative conditions. First of all, on your left, you can see how the combination of gray matter and white matter can improve the diagnosis, the differential diagnosis between Alzheimer's disease and frontotemporal dementia, which can be very challenging in the initial phase of the disease. In the middle, you can see how tractography can help in the diagnosis within the frontotemporal dementia spectrum in differentiating different clinical phenotypes. And on your right, you can see that you can also combine very nice language task, experimental language task that are used in patients with aphasia with some information obtained using both voxel-based morphometry and tractography, and how the model, the combined model combining language plus MR features is the best model in predicting the clinical diagnosis. Finally, we tested whether functional connectivity can also help in making the right diagnosis. And we first look at the classic network connectivity within the main networks in the brain, such as the hormone network and the salience network. And we show that this information can help the clinician in differentiating the 2 main dementias, so the Alzheimer's disease, which is associated with the involvement of the posterior brain networks, and the frontotemporal dementia, which is associated with an opposite pattern, so a functional connectivity decrease in the anterior brain networks. And then we also move from standard functional connectivity approaches to connectomics and graph analysis, and again a couple of examples of how these approaches can be useful in understanding better the differences between these two conditions. So again, Alzheimer's disease associated with the parietal and temporal regions involvement, and the frontotemporal dementia associated with the loss, for instance, the loss of cortical hubs in the frontal and temporal regions. But making the right diagnosis in neurodegenerative conditions means also to be able to predict the underlying pathology, as an important issue in these patients is that the same pathology can be associated with different syndromes, and also the opposite. So the same syndrome can actually be associated with different pathology. So how MR can be useful in helping the clinician in predicting pathology? Here an example in the frontotemporal dementia spectrum. So we now know that different pathologies within this spectrum are associated with, can be associated with different patterns of atrophy. And using a standard VBM approach, you can try to guess which is the underlying pathology in your patient. And we also look at this issue in a particular group of patients, which are patients with primary progressive aphasia, which can be associated with both frontotemporal dementia pathology and Alzheimer's disease pathology. And we suggested that looking at both gray matter and white matter pattern of damage can improve the diagnosis, not only of the clinical diagnosis, but also of the underlying pathology in these patients. The second major issue is not only to associate correctly the syndrome with the pathology, but also to understand which are the correlates of the genetic heterogeneity in these patients. I'd like to show you this example from patients with amyotrophic lateral sclerosis. ALS can be associated with different genetic mutations. The most important is a repeated function in this gene, the C9orf72. And carrying this mutation is associated with a more rapid disease, with more frequent cognitive and behavioral symptoms in these patients. And looking at both cortical thickness and functional connectivity and atrophy in the subcortical regions, for instance, we show that these patients, so those with this mutation, are characterized by a specific subcortical damage within the thalamus and the cerebellum, with a particular pattern of occipital functional connectivity. And again, we did the ALS group patients, looking at another genetic mutation, which is SOD1. We show that these patients are more associated with cervical cord damage compared with the sporadic patients, and so on. So again, MR can be useful not only to make the right clinical diagnosis, not only to help to guess the underlying pathology, but also to understand better which are the structural and functional correlates of genetic heterogeneity in these conditions. Another major point in neurodegenerative conditions, as I told you at the beginning of my talk, is not only to make the right diagnosis at the beginning of the disease, but also to be able to predict the progression of the pathology in the CNS of your patient. So predicting the progression of pathology means to be able to predict the clinical evolution and survival in these patients. And I'd like to show you this recent work from our group again, looking at the prediction of clinical evolution and survival in amyotrophic lateral sclerosis. That is a very rapid disorder. You should consider that the median survival of patients with ALS is around 4 years. So we obtain at the time of onset of the disease 3T MR scans in 200 patients with ALS. looking at both gray matter and white matter degeneration, not only within the motor network, but also within the extramotor networks. And then we follow up our patients for many years, and we try to explore which is the best multivariable model for survival prediction in our patients. And when we look only at the clinical characteristics of these patients, our prediction was good, but we obtain a better prediction of the survival when clinical characteristics, cognitive characteristics were combined with the gray matter and white matter features. And here you can see the clinical multivariable models. Here the combined model. In the all patients, we were able to identify three groups of patients according to the survival, compared with the two groups that were obtained using only clinical variables. The other major points, so making diagnosis, predicting the clinical evolution, but we would also like to monitor the progression of neurodegeneration. So we would like to provide biomarkers to test, for instance, the efficacy of drugs in these patients. And we are trying to follow the progression of our patients, obtaining scans every 6 months. So in order to obtain a big longitudinal data set, so I'm going to show you some results in specific groups. Here, for instance, results obtained in a group of patients with atypical Parkinsonism, so patients with progressive supranuclear palsy. Here you can see the pattern of atrophy at baseline and the pattern of white matter damage at baseline. So nothing not expected. So these patients are characterized by atrophy in the frontal regions and also by microstructural damage of white matter. What happens after 1.4 years in our subjects? Very subtle changes in the gray matter, while if you look at the white matter degeneration, we were able to show a widespread accumulation of damage, suggesting that maybe the gray matter assessment is very useful at the initial phase of the disease, but white matter damage, the assessment of white matter damage is more sensitive to accumulation of changes over time in these conditions. And similar findings in patients with Parkinson's disease. Again, we obtained serial MR scans in a large group of patients with Parkinson's disease with different disease stage. So we use a cluster analysis in order to separate patients in different clusters according to many clinical and cognitive features. And here you can see that at baseline, gray matter atrophy was present in patients with moderate to severe Parkinson's disease, while it was absent in patients with early Parkinson's disease. But when we look at the accumulation of atrophy over 4 years, so cortical thinning was present in patients with the early Parkinson's disease more than in patients with moderate to severe Parkinson's disease. So suggesting that MR can offer you different biomarkers, and you have to choose the right biomarkers according to the stage of the disease you are studying. So if you are looking at the early phase of the disease, maybe cortical thickness and atrophy is the right biomarkers. When we are looking at patients in the moderate to severe stage of the disease, maybe it's better to look at the neurodegeneration within the white matter. And finally, we are trying to make the situation even more complicated. So we are trying to apply connectomics in order to predict the spreading of pathology within the brain of our patients. The idea is behind this approach is the so-called prion-like hypothesis in neurodegenerative conditions. So the idea in neurodegenerative conditions is that pathological proteins, which, as I told you, accumulate in the brain of these patients, are able to move from one region of the brain, which is the epicenter of the disease, to other regions using wild matter tracks and connectivity as the way to move from one region to the other one. If it's true, we expect that this spreading is proportional to the path between the epicenter and the other nodes, but also is proportional to the strength of connection within the epicenter and the other nodes. And so we can expect that when the pathology involves, for instance, one connector hub, the pathology can spread from one network to the other one. So we try to we are trying to test these hypotheses in different conditions. Here, the first example we obtained in Alzheimer's disease patients. So on the left, you can see, as expected, the pattern of atrophy in patients with Alzheimer's disease and in patients with mild cognitive impairment. Nothing very special. Widespread pattern of atrophy in patients with overt dementia, a relative focal atrophy within the parietal, temporal, and frontal regions in patients with MCI. So the most atrophic region in both Alzheimer's disease and mild cognitive impairment patients was the hippocampus. So we use the hippocampus as the epicenter of the disease. And then we move into the brain of healthy subjects when we look at functional connectivity with the hippocampus. And what we found is that there is a correlation between the distance between regions of the brain and the hippocampus, but also between the strength of connections between regions of the brain and the hippocampus and the degree of atrophy in the brain of patients. So shortest is the distance between a region and the hippocampus. Highest is the connection between the region and the hippocampus. Greatest is the atrophy of that region in patients with Alzheimer's disease, suggesting that the path and the strength of connections are two features able to modulate the progression of the disease. So in conclusions, I show you some examples in conditions that have a high disease burden. They are all characterized by, they can include young onset patients, treatment side in development. We don't have reliable outcome measures, and so the high healthcare costs are obvious in these conditions. I try to show you that MR can be used to provide reliable markers for neurodegeneration, not only to make the right diagnosis, but also to predict and monitor the conditions. This is possible not only in the most frequent neurodegenerative conditions, which is Alzheimer's disease, but also in other conditions. And of course, the aim is to make the right diagnosis, make the right prediction in order to provide personalized therapies in this patient. Thank you for your attention.